Today's topic is about ammonia and metabolism of amino acid. Simply that metabolism of amino acid ultimately forming ammonia, and ammonia that is important molecule and it is very dangerous for our body. How it is generated due to this metabolism of amino acid that we will look in this video. So first about metabolism of amino acid. Now simple that metabolism including both the thing anabolism plus catabolism. Anabolism means making of molecule, catabolism means breakdown of the molecule. So anabolic reaction in protein metabolism where protein are synthesized from the amino acid. The catabolic reaction where dietary protein and body protein are broken down to the amino acid level. So further from this amino acid ammonia will be generated as a process of the deamination reaction, and for that particular reaction transdeamination reaction is needed to remove amino group of this amino acid and forming ammonia, and finally to excrete this ammonia in the form of urea. And for this transdeamination reaction is needed. This word including two reactions, transamination plus deamination. So the carbon skeleton is used for the synthesis of the non-essential amino acid also. So this reaction is also have some biological importance that also we will look in this video. So it is also used for the gluconeogenesis and for the complete oxidation to provide the energy. So that is including in this metabolism of protein and in relation to the ammonia formation. So first that formation of ammonia. The first step in the catabolism of amino acid to remove this amino group of amino acid in the form of ammonia. This is the major source of the ammonia. So basically this ammonia is generated due to that metabolism of amino group from the amino acid. However, small quantity of ammonia may also be formed from catabolism of purine and pyrimidine bases. Now, ammonia is highly toxic, especially for the brain. Detoxification of ammonia is by conversion of urea and excretion to the urine. So, main purpose or target of this ammonia detoxification that is to form urea and excrete to the urine. Now, how exactly it is occurring? So, first reaction that is transamination. We have seen that transdeamination, which is including two reactions, transamination plus deamination. First detail about the transamination. Now, what is the meaning of that transamination? It is the exchange of alpha amino group in between one alpha amino acid and another alpha keto acid, forming a new alpha amino acid. Simple, you have to remember one amino acid is there, and keto acid means simply it is containing C double bond O group. So that one amino acid combine or react with the alpha keto acid. So what happens? This amino acid will be converted into the another form of the keto acid. This amino acid is now converting to the keto acid. Now this keto acid will accept this amino acid, and this keto acid will further convert into the another form of the amino acid. Now this reaction is most important basically to form an important molecule that is glutamic acid. Why? That also we will look in this video. So amino group is interchange between different amino acid, that alanine and glutamic acid, but particularly for that pyruvate molecule or alpha ketoglutarate molecule is needed. So in next reaction that you will understand how exactly it is occurring about transamination. So in almost all cases amino group is accepted by alpha ketoglutric acid, which is important intermediate molecule of the Krebs cycle. So that alpha ketoglutarate will be converted into the glutamic acid and it is generated. So enzyme that is known as a amino transferase. Now this enzyme which utilize for this transamination, for that activity pyridoxal phosphate, that is vitamin B6, is needed as a prosthetic group or coenzyme form. So it is readily reversible reaction, that is transamination. So in detail of this transamination that we will look. So simply what we have seen, amino acid plus keto acid. So it is interconversion of this amino group and keto group in between two molecules. So simple that glutamate and pyruvate. Pyruvate that is also end product of the glycolysis process. So this pyruvate is containing keto group. Now over that keto group, this amino group from this glutamate is transported. So this glutamate is now will take up this keto group from this pyruvate. So it is now converted, glutamate is converted into the corresponding keto acid. So glutamate is making alpha ketoglutarate, while pyruvate which is getting this amino group, now it is converted into the alanine molecule. So simple that reaction, glutamate is converted into alpha ketoglutarate that can be utilized for the particular Krebs cycle. Pyruvate which is now converted into the alanine, this alanine that can be also converted or diverted for the gluconeogenesis if it is needed. So that is interconversion, and for that pyridoxal phosphate coenzyme is needed, that is vitamin B6. Same way this reaction is reversible. So alpha ketoglutarate can combine with the alanine and can form glutamate. Same way pyruvate will be generated. So that is reversible reaction, that is transamination reaction. Now, what about biological significance of this transamination? So, it is considered first step of the catabolism of protein. So, the ammonia is removed and the carbon skeleton enters in the catabolic pathway. Synthesis of non-essential amino acid. So, all non-essential amino acid can be synthesized by the body from keto acid by this transamination reaction. Those amino acids which cannot be synthesized in this manner are therefore essential and they should be made available in the food. So simple that for the formation of non-essential amino acid, transamination reaction is utilized. Now for the interconversion of amino acid, if one amino acid is high and the other amino acid is low, then the amino group from the one amino acid is transferred to the keto acid to give rise a second amino acid or to equalize the quantity of both. So that is interconversion of amino acid is important reaction, and for that interconversion basically purpose to maintain amino acid level or all amino acid in the blood. So this is called equalization of quantity of non-essential amino acid in the body, that is possible with the help of this transamination reaction. So that's all about the biological significance. Next, that is clinical significance. AST and ALT, that is aspartate aminotransferase and alanine aminotransferase, are induced by these glucocorticoids which favors the gluconeogenesis process. But these two are the ultimately transamination reaction enzyme. So these two are the important enzyme and they have a particular clinical significance. Here that about enzyme AST, that is aspartate. Aspartate means aspartic acid amino acid. ALT means alanine amino transferase, that alanine that is also a particular amino acid that already we have seen in previous example. So AST is increase in myocardial infarction while both AST and ALT are increase in a liver disease. So that is particular clinical significance of the this transamination enzyme. Simple that AST, another name of that AST, that is SGOT, and another name of ALT, that is SGPT. So many times as a clinical practice, investigation of this SGPT and SGOT is routinely utilized to diagnose about myocardial infarction and liver disease. For the transdeamination reaction, so amino group is released by coupled reaction of this transdeamination. So first we have seen about the transamination reaction, and that will be followed by the oxidative deamination reaction. So transamination take place in the cytoplasm of all the cells of the body. So that transamination is occurring most of the body, but most of the body cells, so all the tissue is undergoing this transamination reaction. So what happen, amino group is transported to the liver as a glutamic acid, because ultimately alpha ketoglutarate that is converted into the glutamic acid or glutamate, which is finally oxidatively deaminated in mitochondria of the hepatocyte. So this transamination is occurring in almost all tissue, while deamination reaction will occur only particularly in mitochondria of the hepatocyte or the liver cell. So these two components of reaction are physically far away, because one reaction transamination occurring in each and every tissue, while deamination will occur only in the mitochondria of liver cell, but physiologically they are quite correlated to each other. So hence this term transdeamination is utilized for the explanation of the ammonia formation. Now oxidative deamination of this glutamate, so only liver contains this enzyme glutamate dehydrogenase, which deaminates the glutamate or the glutamic acid. So all amino acids are first transaminated to glutamic acid, which is then finally deaminated. Amino acids are deaminated at the rate of 50 to 70 gram per day. So that much amount of ammonia removal is occurring inside the mitochondria of liver cells. So hence the glutamate dehydrogenase reaction is the final reaction which removes amino group of all amino acids. Simple amino group that you have to remember, that is NH2, which is present over the amino acid. Now for this reaction, NAD plus or NADP is needed as a coenzyme, and it is an allosteric enzyme which is activated by ADP. So when there is a low energy level, this enzyme activity will increase, while inhibited by the GTP. GTP is indicating high energy level. During that time this activity will be decreased. Now further in detail of this particular transdeamination reaction, transamination simple, that is any amino acid that is converted into the corresponding keto acid, and most commonly utilized, that is alpha ketoglutarate, which is forming glutamate. This reaction is occurring in all tissue of the body. Now this glutamate is transported to the liver. So first transportation of this glutamate to the liver from all tissue. Now finally this glutamate is deaminated in the liver mitochondria. and for that enzyme glutamate dehydrogenase is needed, and for this reaction NAD plus is utilized. So NAD plus is converted into the NADH, and glutamate dehydrogenase enzyme will convert this glutamate into the alpha ketoglutarate, and ammonia molecule is generated. So that is one important reaction where ammonia is generated in the body. Other important or minor pathway for the deamination or ammonia generation that also you have to remember, L-amino acid oxidase. It can act on all amino acid except hydroxy and dicarboxylic amino acid. Over that, this enzyme is not working. Basically, this L-amino acid oxidase uses FMN, that is flavin mononucleotide, as a coenzyme. The peroxide form in this reaction of L-amino acid oxidase is decomposed by catalase enzyme in the peroxisome. Another minor pathway, that is D-amino acid oxidase. It can oxidize glycine and particularly D-amino acid, which is normally not present in the body, that may be formed due to bacterial metabolism. So all will be deaminated with the help of D-amino acid oxidase. Here for the D amino acid, you can remember that FAD as a coenzyme. For the D, FAD, and for the L, that you can remember FMN. Now, monoamine oxidase, that is another enzyme. Ammonia may be formed in the body through minor reaction like oxidation of monoamines by monoamine oxidase enzyme. So that three are the particular minor pathway for the ammonia generation. Now, non-oxidative deamination. So sometime this oxidative deamination that we have seen, that transdeamination reaction, now sometime non-oxidative deamination, particularly dehydratase, act on hydroxyl amino acid to remove ammonia. Which are the hydroxyl group containing amino acid? Serine and threonine. So that will undergo to this enzyme dehydratase. So serine is converted to the pyruvate, threonine is converted into the alpha ketobutyric acid and making or releasing of this particular ammonia molecule. So that is called non-oxidative deamination. Same way desulfhydratase, so that is also acting over this sulfur group containing amino acid, that is cysteine. So that is also converted into the pyruvate. During that time also ammonia will be generated. Histidase, so histidine is converted into the urocanic acid by removing that ammonia amino group of this amino acid. So ammonia may also be produced by gastrointestinal tract by bacterial putrefaction. So some bacteria are already present in gastrointestinal tract. When they are metabolized this particular protein, sometime that ammonia will be generated, that is called bacterial putrefaction. So these are the other important reaction where ammonia is generated. Same as a amino group, you have to also remember about amino acid, that carboxyl group, that is also important part of the amino acid. So if suppose carboxyl group is removed, and that process that is called decarboxylation, it is also important biologically. The amino acid will undergo alpha decarboxylation to form the corresponding amine. Simple histidine is converted into the histamine, tyrosine is converted into tyramine, tryptophan is converted into tryptamine. These three are also important. One of the most important, that is histamine, that is used as a allergic mediator inside the body. Simple amino acid, there is a structure, there is a R group, carboxyl group, amino group. Carboxyl is removed in the mostly in the form of that carbon dioxide. So corresponding amino group is, or amine group is generated from this amino acid. So that important decarboxylation reaction. Now finally, detoxification of ammonia. So simple amino group, that is NH2, ammonia, that is NH3. So this detoxification of ammonia, that is considered the first line of defense. Because this ammonia is highly toxic, ammonia should be eliminated as and when it is formed inside the body. Even very small quantity of this ammonia may produce toxicity in the brain. So ammonia is produced by almost all cells including neurons as a reaction of this oxidative, particularly that deamination reaction or minor pathways of this deamination, and this ammonia that is ultimately detoxified in the form of urea. But this ammonia, which is produced by almost all cells, mostly trapped by this alpha ketoglutarate to form glutamate, as well as suppose that glutamate molecule is present, then glutamate will convert into the glutamine to trap the ammonia molecule. Now intracellular ammonia is trapped by the glutamic acid to form glutamine, and mostly this process is occurring in the brain to avoid toxicity of the ammonia over the brain. Now glutamine is transported to the liver where the reaction is reversed by the glutaminase. So glutamine is transported finally to the liver, where this reaction of reverse means glutamine is reconverted to the glutamic acid or glutamate, and that process will occur with the help of glutaminase enzyme. Now aspartic acid may also undergo the same reaction, that is aspartic acid will convert into asparagine. Same way this asparagine will go in the liver and reconvert it into the aspartic acid. So that is important disposal or first line defense of the ammonia, for because this ammonia is highly toxic, so it need to be detoxified. So simple, what happen, glutamic acid will combine with the ammonia and that will be converted into the glutamine, so that is glutamine synthetase. Here that ATP is utilized, so it is synthetase. Same way glutamine will reverse back to the glutamic acid or glutamate with the help of glutaminase, this enzyme present in the liver. So these two reaction is also important to trap this ammonia and release this ammonia in the liver. So finally this ammonia will be converted into the urea molecule. Further, this glutamic acid or deamination reaction, amino sugar that is also providing this ammonia, pyrimidine, putrefaction in the intestinal tract, glutamine and asparagine, that all are mostly providing ammonia and making ammonia pool. Now this ammonia that is generally react with the glutamic acid and making glutamine. Same way it is acting with the aspartic acid and make asparagine, and most common thing that is disposal of this ammonia in the form of urea to excrete in the urine. Now the transport of this ammonia inside the cell, transamination produce glutamic acid, but the final deamination and production of ammonia is taking place in the liver. So glutamic acid act as a link or transporter for this ammonia group, so between the amino group and the amino acid and ammonia. So simple, the transporter of amino group, that is with the help of glutamic acid. So concentration of glutamic acid in blood, that is 10 times more than the other amino acid. Simple, we have seen that roughly all amino acid is 1 milligram per deciliter, while glutamic acid means glutamate and glutamine, these two are particularly 10 times more than the other amino acid. So glutamine is the transport form of ammonia from brain and intestine to the liver, while alanine is the transport form for the muscle. Another purpose of this alanine for the gluconeogenesis also. Now final disposal of this ammonia from all over the body is detoxified to form urea by liver cell, and that excrete to the kidney. So this final disposal will occur with the urea cycle. So urea is the end product of the protein metabolism as well as final disposal of the ammonia. So you have to remember, end product of protein metabolism, protein is degraded to the amino acid level. Amino acid will release amino group in the form of ammonia. This ammonia is highly toxic, it need to be converted into the urea, and that will occur only in the liver with the help of urea cycle. So that's all about the metabolism of amino acid and ammonia formation, and finally this removal of ammonia from the body in the form of urea. So that's all about today's lecture.